Okay, thank you very much for having me here. Given that the last time I checked, there was 16 inches of snow in Boston, I'm very happy to be here today. 16. Yeah, Providence got 33 inches of snow. So better. So Boston's doing better than Providence, or Anyway, I'm going to talk to you about the general topic is cancer cell plasticity. I'm going to talk to you about in the context of drug persistence, and I'll define that precisely in a moment. But this idea has many more other ramifications, which I'll try to talk about at the sort of the second half of the talk if I get there. But if not, that's also fine. This is work, we published our original version of this a couple of years ago, and there's a new version of this that's actually still in preparation with a student of mine. Okay. Bacterial persistence is originally the idea that if you provide antibiotics to a bacterial colony, most of the cells will die, and they'll die at some particular rapid rate, sort of that line going down over here. Whoops, yeah, there we go, over there. But there'll be a subpopulation of cells that will somehow be more able to cope with that antibiotic, and they will grow, they will decay at a much slower rate. That's what that curve over there. And this was identified really in the 40s by Biggers as an interesting phenomena of how bacterial systems respond to antibiotics. Now, there's the classical picture was always this sort of two-step decay here, and the second step is, there's two possibilities. One is that these cells are actually dying just at a much slower rate. The other possibility is that there are ongoing transitions from whatever this cell type is to whatever that cell type is, and this lower rate is just reflecting the transitions back to a sensitive cell type, which then dies at a rapid rate. So either way, it gives you the same two-state things, and people have studied this for a very long period of time. Starting about, 15 years ago, maybe a little bit longer, people have started noticing that similar things occur when you provide therapy to cancer cells. And we'll talk about some experimental examples in a moment, but originally the idea is that this should somehow be completely analogous to what happens in bacterial cells. So in bacterial cells, the idea is there are pre-existing multi-stable networks that can give rise to different phenotypes of the bacteria, and those different phenotypes have different sensitivities to the drug. When you provide the drug, you're doing a selection experiment. You're not selecting genetic variations, you're selecting phenotypic variations. But aside from some very important differences, for example, the idea that these can be reverted back to the sensitive type, because after all, this is not a genetic change where reversion is very, very difficult to imagine. This is a phenotypic change, it's a change in the state of the system, and you can imagine going backwards also in terms of a phenotypic transition. So then it would be modeled by the standard, Luria-Delbruck type ideas, and everything would be in parallel with what was known for bacteria. And I guess one of the first points of what I want to make in my talk is we actually don't think this is sufficient for the cancer system. We think this is much too simplistic for the cancer dynamics of how persistence arises, and that's what I will tell you about where we came to that idea and how we think about the system in a more complicated way. So we came to this problem a few years ago, maybe 5 years ago, when we noticed this particular paper, and this paper is focusing on we already heard about non-small cell lung cancer. This is focusing on EGFR-driven non-small cell lung cancer, which means that the receptor EGFR is overexpressed, and this drives uncontrolled growth, and therefore, if you can apply drugs which block the activity of the EGF receptor, in particular some type of antibody, you can use that as a targeted therapy for this particular type of lung cancer. So this was a typical experiment which looked at this idea of persistence. Now, persistence means, again, that it's not resistance. It's not that you have a mutation that prevents a drug from binding your target. It's not that you have some other version of a genetic change. It's believed to be a change that happens at a constant genome. It's a change purely in the phenotypic state of the cell, what the cell is actually doing with its existing genome. And here, and this was one of the original systems, this EGFR resistance, which demonstrated this possibility, and these people tried to understand the question of what percentage of your population would be in this persistent state. Is it 1%? Is it 10%? How does that depend? So what they did is they took lung cancer cell lines, and they created from it single cell clones. So clones doesn't mean, again, genetic differences. It means just different cells that are picked out one at a time from the population and then grown up as all the progeny arising from a single cell. And they tested for those different single cell clones what was the number of what was the percentage of the population that was in this persister state, that when you applied this drug, you would get most of the cells die, and this percentage would survive. And their definition of survive is ones that were still present at day 7 after you give the drug to the system. That was just their definition of something they called the chance to persist. And what they discovered is that this is a number that is what I can think of as a quantitative trait of the of the cancer system. So here is a plot of a distribution of all their different clones. So each of their circles is a data point from a particular clone that then grows out into a population emerging from a single cell. And each of them has a different value of this parameter. So this parameter down here is the percentage of cells that survive after 7 days of treatment. And it's a distribution. And so what we wanted to understand first is what determines this distribution. Can one understand what's going on over here? But the first thing it shows you is that different clones, which somehow are different from each other, are giving rise already to very different numbers of this. It's not like your whole population has a fixed percentage of a particular type. This number is something which depends very strongly on some details that are going on in your system. So first, let's talk about the simplest model of this. The simplest model of this emerged from looking a little bit more at their data. So here is their data of 2 different examples of cell lines, PC9, G6, whatever. And these are the growth, effectively the growth rates of those things as a function of this parameter CTP. So CTP, remember, is the chance to persist. And this says there's an inverse relation. It's pretty noisy, but the inverse relation says if your clone has a greater percentage of chance of persisters, it means that your growth rate in the absence of any drug is slower. So there's a negative selection for chance to persist in this system. So you can make a very simple evolutionary model where all you do is say, okay, I have a parameter X. My population depends on this parameter X. And I have a selection term which says that increasing X is a negative term in your selection. You also have something that allows dynamic changes over long periods of time in this parameter, because after all, this is not a genetic parameter. This is, we're not looking at mutations. This is just some type of phenotypic drift of the population. And you can write down the simplest equation. The simplest equation has a steady state solution, which is just an Airy function, for those of you who are not into special functions. But nonetheless, this immediately fits the data. We can look at the red line. This is a fit with basically one parameter. Well, sort of really one parameter. The 17 here is the only parameter you have to fit. That comes at some dimensionless ratio that you create from the rate of diffusion in this space and the rate of growth decline as you increase X, and you can fit the data, basically as well as you're ever going to fit any cancer data, as far as I'm concerned. Yes?
Just why don't you call the 0.06?
Say again?
The 0.06, why don't you call that a parameter?
So 0 is strange because 0 is an end of this system. So you really have to decide what you're going to do about the boundary conditions at 0. The boundary condition is also at 1, but there the population is 0. So you can put in some type of boundary layer which will match the boundary condition at 0 if you want. This is just the simplest analytic solution. You can do better if you really impose some boundary condition that there's no flux out of 0. You can solve the problem also, and it's just more complicated. But it doesn't change much. It just changes it very close to 0, and you don't have any data there anyway, basically. Okay, that's just the technical point. Okay, now, so then if you're a biologist, and then this is great, it fits the data, but the question is, what is X? What is determining this particular macroscopic parameter that is being shown as a distribution? And so our perspective has been, let's go to the next slide, is that X has to do with the epigenetic degrees of freedom of the cells. Now, for those of you who are not into biology per se, let me just give you the 30-second introduction to what we mean by epigenetic degrees of freedom. And the answer is that, this is a picture, schematic picture of what the DNA looks like in your cell's nucleus. It starts out as a double helix, but then, as double helix, would not fit directly into your nucleus. It's just too long and too, even wrapped up into some ball, it's too big. So you have to package it in some way. And when you package it, you wrap around these things called histones, which form these things called nucleosomes, and then these sort of pack together, and then eventually you come up to this larger scale, and you see something recognizable as a chromosome, in the traditional view of a chromosome. So it turns out that this whole packaging steps over here allow you to modify how accessible the DNA is to the machinery that makes the proteins for your cells. Here is, again, a schematic picture of what one of these little balls look like. It's actually 8 different proteins called H3A, H2A, H4, whatever. All these 8 things is sort of a protein complex made. Those proteins are called histones. Each of those histones have long tails. Those tails can be modified by adding various chemical groups to them. They can add methyl groups, you can add acetyl groups, you can add other groups. And when you make those different modifications, you change how tightly the DNA is wrapped around those things. You change how easy it is to kick off the DNA from this spool versus not kick it off. And all those things affect how easy is it to access that piece of DNA to actually make the proteins you need in your cell. So at a very rough level, various changes in these what are called histone marks, just these different chemical modifications of these tails of these proteins, change the degree to which you can, for example, start expressing a new gene in your particular cell if the circumstances, the environment of the cell somehow demands that that new gene be expressed. It's a sort of way in which the cell can decide to be very non-plastic. If you have a very differentiated cell type, it has very specific ways in which large portions of your genome are basically tucked away in your closet and never used, and very hard to access at all. On the other hand, if you're a more adaptable cell, a more stem-like cell, if you want to use the language we heard before, then major portions of your DNA have to be accessible because your stem-like cell doesn't yet know what it's going to make. If it's a general purpose progenitor cell, it can make T cells, B cells, NK cells, all from the same progenitor. It has to have much more accessible DNA. So the general idea is that the accessibility, as determined by these marks, is directly related to how adaptable the cells are going to be when faced with some signals telling them what they need to do in order to survive. So this X parameter is what we believe is related to this, and there's a whole bunch of evidence.
Sorry, very quickly ask, these histone tails,
yes,
they are not genetically defined, right?
No, they're post-translational modifications, right? So they're phosphorylations, acetylations. It goes on, many different choices, all done as chemical modifications. It's not genetic. I mean, of course, there are enzymes that make those things that have different genetic expression levels, but those are, in our system, those are just fixed things. Okay. So, not only does an argument sort of always made senseless, but if you look at the literature, what you begin to notice is that, many of the papers that focus on these types of persisters have already noticed that this is really an important consequence. This is, in some sense, one of the first papers on the subject, chromatin-mediated reversible drug-tolerant state, and you notice already the title focus on chromatin mediation of this phenomena. And if you read the paper in more detail, you discover statements like this ability to create persistence depends on particular enzymes that modify these tails in order to make the DNA more accessible. So here is one paper. This is from this is already old paper. This is a new paper that just came out this year, single cell resolution of an open chromatin signature in persister tumor cells. So it's the same idea that the persister cells, the ones that are somehow able to adapt themselves and survive in the presence of these drug attack, are the ones that somehow have this much more open signature, and that enables them to express new types of genes that they would not necessarily be expressing in an attempt to overcome the drug. Your question, yes?
Do you think they already have the chromatin more open, say they are more prone to be more plastic, or is it a consequence of the drug?
So I'll get there. So I think the answer is both can be possible. Certainly it can be possible that cells are pre-existing, and there's evidence for that. But there also is some groups that have provided evidence that that can be induced by the drug.
So for even the same subtype, right?
Yeah. Go ahead.
And how static is that? I mean, these epigenetic marks, they are also changing over time.
Yeah, so that's the line in the bottom. Epigenetic marks are heritable for some time, but can eventually drift. I was just going to say that next. So the answer is they're somewhere in between the things we usually think of as genetic circuits, which are due to proteins and mRNAs, and they decay. Maybe the time scale of those things, maybe hours or days or whatever. And these things are sort of on the time scale of maybe weeks to months. So they are inheritable at some level, but much less fidelity than typical genomics.
Especially in cell division, they are lost and then they are...
So there's a whole field of endeavor that tries to figure out how the cells manage to use the existing marks to recreate the same marks during a cell division when you have to insert all sorts of new histones. So there's a whole literature on people trying to create models of that, and it's obviously not a perfect system because they do drift on some time scale. Okay, so the idea that adaptability is somehow connected to these different chromatin states is not an idea that's only emerged in the context of drug persistence. Because Eduardo was sitting here and I knew he was giving the next talk, I would just refer to a paper we wrote together a couple of years ago showing that if we look at some other aspect of cell showing adaptability, and for those of you who don't know what EMT is, that's another whole long hour talk that I decided not to give. But it's basically some way in which the cells change from epithelial-like, which means they stick to their neighbors and stay put, to mesenchymal-like, where they don't stick to their neighbors and migrate. The ability to do that change is also very closely connected to epigenetic factors, to these chromatin factors. And that's, I'll be happy to tell anybody about that in gory detail. It's, after my talk. Okay, so this idea, yes, go ahead.
I just had a question about, so you talked about the different time scales with the epigenetic. Can you measure these things the same?
Yes.
You can measure the same epigenome and the same cells over time, or is it you have to?
It's hard to measure it over time. I mean, just like RNA transcriptomics, it's hard to measure over time. You can measure it at a snapshot where you can get a full, the way it's usually measured these days, you can look for specific histone enzymes and see where they sit on the genome. The more I don't know, simple way or the way people are really doing it these days is something called a TAC-seq. A TAC-seq is where you have some type of transposon factor and you see which parts of the genome are accessible to it. So you can get a genome scale map at not very great resolution of which parts of the chromosome are accessible and which parts are not accessible. Now, it used to be that you had to do that, so it followed, sort of 10 years after the RNA transcription story, because it used to be you did that in bulk. And then people invented single cell. And now, over the last couple of years only, you can take the same single cell that you're doing transcriptomics in, and you can measure this ATAC-seq. So you can get, in the same cell, a measurement of the chromatin structure at some coarse-grained level, plus the actual transcription. And the bioinformatics community is rapidly trying to develop technology for helping to interpret that data. So that's relatively new, over the last couple of years only. So yes, you can measure these things in either very focused things with higher resolution or a very general sense with weaker resolution on the genome scale. Okay, besides just trying to fit that equilibrium distribution, we wanted to understand various other things about this process. So in particular, we wanted to understand this issue of the cells dying after, continuing to die after the first 7 days. So, the picture looks sort of like this Bigger's picture in bacteria. You have a rapid decay for the first time, and then afterwards you have slower decay. And what they noticed in this paper was that different amounts of persister percentage gave rise to different decay rates. So it isn't like there's a unique decay rate, that all these different clones have different decay rates, and they're and their order of the decay rate goes along the inverse order of the percentage. So the higher percentage you have of persisters, the slower this thing decays as a function of time. So it's not like there's a single number giving you a decay rate. You have a distribution of this x parameter, and then that also feeds into the distribution of the death rate. So we extended our model to try to figure out why are these cells dying, and that actually led us to introduce another continuous variable. So it was bad enough that we had one PDE in one variable, but now we're going to have a PDE in two different variables, because the answer is that even if at a fixed clone, X is a label of a clone, you still have cells that are behaving differently. Some of the cells die and some of the cells don't die. The percentage depends on this X, but the individual cell still has its own degree of survivability. So we introduced another parameter, S, which we call the survivability. And the way it works, I'll show you in the next slide, is that you have a distribution of X. The distribution of X is set by this X parameter because it tells you how many survive till day 7. That was the definition of my chance to persist. And then the ones that persist are down at this edge over here of the distribution. So you have some type of Gaussian distribution centered around some S naught. The ones that are negative die, the ones that are positive don't die, and the way in which those deaths take place in that slow regime is cells diffuse from regions where they're able to survive from positive S's to negative S's at some rate. So you can calculate that rate in our model. You can calculate it analytically pretty much. You can certainly do it numerically, and you can work out all that. And what we got was that curve over there. I said this is partially analytic, partially numerical for all those different things. I'm not worried about the quantitative agreement. You can fit the data. But what we were most happy to get is this qualitative assessment of how those different decay rates were ordered in terms of how much persistence you actually had in the population. So we got exactly the same ordering, and then we could actually fit the data if we wanted to. Yes?
Really dumb question.
Please.
The drug, when you say administered, you mean it's continuously?
Continuously administered, yes. Continuously administered in this case. Okay. Now, what is missing from our picture, right? I mean, it's a great picture, but what was missing? Why did we write one paper and now we're writing another paper? And the answer is because there are phenomena that existed in the general literature that was not in that particular paper that we were analyzing that we wanted to understand. And the most important of those phenomena is the fact that in some other systems, you actually get these persisters at some point transition to a state which is called a cycling persister. So even though you still have, the drug is still there, this is your question, you never stop giving it the drug, but some of the cells, another percentage of the cells that were persisters and so have survived up to that point, now start actually going back into the cell cycle and reproducing. Not as fast as the original population, but nonetheless, they're back able to reproduce. So these were called cycling, in the original paper, they were called DTEPs, I forget what this E stands for expanding, I don't remember. P is persisters, so this is expanding persisters. So, and then I think cycling persisters is a more easily to remember name as I've just proven to you. So cycling persisters has been investigated in a number of different systems, and the interesting thing about it is it occurs spontaneously. You don't remove the drug, you don't do anything special. Some percentage of the cells, again, a small percentage of the persisters are able to restart their cell cycle, and so start growing again. Now, so this was number one. Our model initially didn't have any possibility of growing again. We had these death curves, but we didn't have anything that allowed it to grow again. Another thing is, I think what was asked before over there, which was, in our model, we're sort of still doing selection, but there's numbers of groups, and I think maybe Eduardo's going to talk about this in his talk, I don't know. There's numbers of groups that have argued that the drug can do more than just select. It can actually certainly can induce plasticity, and maybe even induce drift in the direction of things that are more open, which therefore make the cells more survivable. So I view that as sort of as a Lamarckian term in the evolution. But, until now we've been doing Darwinian evolution, and when you start putting in things like drift that's driven by the actual perturbation, then you start talking about things that historically would have been called Lamarckian evolution. Okay, doesn't matter. And this idea already appeared in another paper by a group in France, where they actually also had put in, in some similar context, that also put in a Lamarckian advection term. So yes.
Has it been excluded that there are resistant mutations?
Has it been excluded there?
That there are genetic events in these cells that
yes.
continue the cycle?
Yes.
Can it be experimentally excluded?
Yes. So the first paper there, I think, and then the subsequent paper in 2015 by the same group, I think went to great extent showing that what happens is you get cycling persisters, and then after a while, because now you're reproducing and you have these cycling persisters, eventually they can pick up full mutations. So they showed that the cycling itself is not due to a mutation, and maybe that's, and depending on your view about whether mutations typically arise during cell division or not, you might guess, well, it's hard to mutate if you're just sitting there and not dividing at all, as persisters are not doing. So they show that actually mutations that arose at a later stage from these cycling persisters. Yeah.
Is it understood that the mechanism of this Lamarckian thing is like any signal transduction activation that is?
I don't know if we have a real mechanism. In other words, maybe I'll also maybe mention what I think could be a mechanism, but none of these papers directly measure a mechanism. In other words, the mechanism has to be, what do you sense about the environment that somehow induces you to become more plastic? Okay, I'll tell you my speculation about that in about 5 minutes or so when we get there. Okay, so anyway, so we have an extension of the model. I don't want to, it's more complicated because now it has an advection term, now it has death terms, and it also has a growth term. And the only thing I want to stress is that in model, this is a framework. Don't think of it as a fit model to any data. It's a framework for thinking about the problem. And from this point of view of the framework, you actually can get curves that look like this. This is the survival fraction as a function of time for these different clones. And what it says is if you happen in your population to have clones that have a high persistence level, and that's it's a case-by-case basis, what's the end of that distribution? If you happen to have it, then those cells will last long enough that they'll be able to turn around and start growing again at the end. If you don't have ones at that high level, so there's a critical value for a clone that allows you to begin growing again versus not growing again, and that's going to be a case-by-case basis, which is in fact what you see. In some of the experiments, they never see cycling persisters. In other ones, they do. So this explains, at least in terms of a framework, how you might get something like that given the original distribution of this parameter. And then, of course, this, of course, then wreaks havoc with the notion of what is the best drug concentration to give to population. So even at the level of giving a constant drug, we can also work out, of course, in great detail, drug holidays and the most adaptive strategy of beating this. We can do all that. But even at the level of a constant drug, you find sort of not very intuitive results. So obviously giving a little amount of drug does nothing. That's fine. Increasing the drug then, obviously you do better. But if you increase the drug too much and you believe there's a significant Lamarckian type turn, it can actually start hurting you again because you can actually drive the system to adapt faster. And at some point you just reach either diminishing returns or even turns around and gets worse again if you give too large a concentration of drugs. Now that's a preliminary result because one of the things you have to check is this, of course, is a continuum model. It ignores individuality of the cells. And it's one thing if you turn around after you drop by a factor of 10, but if you turn around after you drop by a factor of 10 to the fifth, then effectively you would just go extinct in any finite in any system of finite number of cells, and that would be fine too. So you have to check that, and we're still in the process of working that out in detail. Okay. Now, I want to mention one new experiment that we have a little bit to say about. This is an experiment from last year. That's part of, if nothing else, this is an experiment that, in the reversal of history motivates what we're trying to do. So this experiment is a group at NYU, what they called resistance continuum in this paper in Nature. And what they did is they have a system where they don't do that. But what they do is they take cells, they have persisters, they give some concentration of drug, the cells persist, they go back into cycling persisters, the system grows back up to confluency in their experiment, and then they increase the drug concentration. And then, of course, again, many cells die, you go into this persister state, cycling persisters emerge, They start growing again, they become confluent again, and then you increase the drug concentration again. So you increase the drug concentration step by step, and you drive the cells from a very, very, no tolerance at all to, in fact, a very, very high tolerance for the drug. And so they have these different tolerance curves, and you see as a function of these individual, cycles of drug regrowth, you're pushing the cells to become more and more resistant, going up some type of continuum. So the advantage of our model is it can explain things like, it can at least in principle make contact with things like this. The thing we've been trying is try to predict, for example, the amount of time it takes each of these recurrences to take place. So in other words, it isn't like you get a constant amount of time. Depending where you are in this continuum, you get either less or more time it takes to grow back up the confluency. So this is, our attempt to fit that data. The red is the simulation, the blue is the experimental data. And this is work in preliminary, and of course, it's an n=1 experiment. They only did this once. So I'm not too worried about detailed disagreements with the theory, but at least we can. The point is not that we can systematically fit some data. The point is that our model can at least make contact with this data because it has that flexibility that you can change the drug concentration, the population will shift, and it will try to react, and it will take a while to do that, to go back into cycling, and you can plot at least how that progression emerges. Okay, in the last. I'm doing pretty well. Last 10 minutes, I want to talk about the broader context. Yes, go ahead.
One question about this one. I don't know if I found something correctly in the previous paper that you were mentioning. Is the just increasing the amount of drug?
Yeah. But they increase it in a very specific way. What they do is they provide some small amount of drug to start with. And you go into this persister phenomena, and it's a small amount of drug, so under small concentrations of drug, you expect and they see cycling to reemerge. And then this is an experiment in vitro, so then the cells grow and they become confluent again in their experimental tissue culture, and then they increase the drug. And they do this 8 times or something like that.
I'm sure you're familiar. I mean, there is this also in vitro experiments with bacteria and antibiotics where they give antibiotics. Yeah. Sometimes what they do instead of increasing them for the resistance bacteria is decreasing. So therefore they create this sort of environment in which the evolution is. Yeah.
So right. So in the drug therapy business, people are very much interested in the whatever metronomic therapy, whatever it's called, and people in Moffitt can tell me what all these things are called, where you try to adjust your concentrations as a function of time to kill as many of the cells as you have. So it's the opposite effect. Here they're trying to train the cells to be more and more resistant. In the metronomic therapy, you're trying to let the cells have enough time to regrow so that the better growth of the sensitive cells take over again, and then you can kill more of them when you restore the antibiotic. So that also is done in the cancer therapy. So yeah, but I don't remember this experiment in the antibiotic. Yeah, right. I don't remember this type of experiment before. This was a rather interesting paper from our perspective because we could at least think about what they were doing. Okay. So this leaves many directions to investigate, and I'm probably not going to get through all of them, but I want to at least make it clear that, okay, this is one problem that arises, but there are many other related issues that one can think about, and many of them people have already asked about. So the first issue I call global versus local plasticity. Our model says cells have some notion based on their epigenetics of the plasticity. And the question is this something pre-existing? Is this something not pre-existing? So this is a different Nature paper, lots of Nature papers in this business. And this is a paper which basically said that there were pre-existing epigenetic factors that actually not only were pre-existing, but actually determined which was the precise way in which the cells would be able to persist. So in other words, what they said is you can go into the original population, you can measure which cells have enhanced plasticity, but the enhanced plasticity is more focused than our model suggests. It tells you not only that the cell can adapt, but it tells you which way the cell can adapt. The way I think of it is which parts of the chromatin are really the ones that are open and able for accessibility to do the adaptation. So they could track this by measuring things. So for example, here is data showing just measure some property of different cells, different clones again, that go through persistence. They have very different, factors of 5 or whatever number that is over there on that axis, factors of difference in invasiveness, just to pick one. They measured many properties, this is just one of them. And they could tell based on the initial state which cells were, which clones were going to do which. So this says that the notion of plasticity really should be thought of as maybe a sort of modular level, that there's, that these ones have plasticity and these genes are available for adaptation, and the cells can find the solution to their drug problem using these genes, and then other clones have different parts of the chromatin which are accessible, and they can use those genes in order to find the solution to their drug problem. And you can tell you can distinguish one from the other by actually looking at which parts of the chromatin are more accessible before you even give the drug. So this is sort of saying at least there's some initial information that is relevant for what is going to happen afterwards. It doesn't tell you what the effect of the drugs are, but this tells you there is some information that is there for at least in this particular case. Okay. Role of metabolism. So somebody asked me where does Lamarckianism come from? Well, besides the epigenetic things that you kept seeing, all those papers also have metabolic factors that seem to play an important role. So for example, in the paper I first talked about, the one about this chance to persist, there are these things on the insulin pathway that seem to be important, that they measure. Again, they could correlate this with these more phenomenological parameters. They could say, gee, if we look at the expression of insulin response factor, and this one correlates best with our. Now they had to do that. This is one of these standard stories in the biology world. These people were very nice. They gave a talk about their data 2 years before their paper was published. And why was their paper published only 2 years later? Because they needed to find a molecular correlate of their measurements. From our point of view, we just were interested in their measurements, but in order to publish it in a high enough level biology journal, they had to spend another 2 years looking for something at the molecular level that somehow correlated. So all they've shown is that it correlates. They don't have a theory as to why it correlates or anything like that, but they showed that this IRS1, which is a insulin signaling response factor, correlated with their measurements of how easy it was to persist. In some of these other papers, for example, in cycling persisters, it's very clear that the metabolism of the cells are playing a critical role in enabling them to start growing again, in particular fatty acid oxidation. All sorts of things. Here is just a review paper about this. And one idea is that maybe metabolism is a way in which you can provide a signal to the cells telling it you're in trouble, something is going wrong, and therefore you should become more adaptable. How does metabolism do that? Well, metabolism has, very strong coupling to all sorts of other things that are going on in cell physiology. For example, all these chromatin marks that I talked about, where you're adding methyl groups or acetyl groups to the DNA or the histones, which is what I really was talking about, all of those use metabolites as parts of the chemical reaction. So if you want to add an acetyl group to a histone, you got to have acetyl-CoA, which is a known metabolite. If you want to add a methyl group, you've got to have some source of methyl groups. Those are all related to metabolites. So if you screw up the cell's metabolism, you automatically affect the epigenetic states of the system. This is well known at a qualitative level. As far as I know, there's no quantitative model whatsoever of how that works. So that's something can be done. We wrote a paper, which is not on this slide, related to this EMT phenomena, this epithelial mesenchymal transition, showing all the way in which it can be linked to metabolism. So if you look at the metabolic system and all the genes there, and you look at all the metabolites, and you look at the genetic network controlling EMT, you notice that there are many, many cross-links between those two systems, which says that cells that undergo EMT can be affected by metabolites and by metabolic enzyme expression rates, and vice versa. Cells that undergo EMT can readjust their metabolism to make use of the fact that now they have a different need in terms of energy usage, for example. So metabolism is an important role for possibly organizing this thinking, which we and other people need to investigate. Number 3, relationship between persistence and dormancy. There's another phenomena in cancer biology, and I'm almost done, don't worry, in cancer biology called dormancy. Dormancy is the fact that this is the most striking case is hormone-positive breast cancer. So breast cancer comes in a variety of types. Hormone-positive means that the growth of the cancer cells are being driven by estrogen or other hormones. And hormone-positive breast cancer has this very unfortunate ability to have cells leave the primary tumor, go into other parts of the body. Typically, the part of the body it goes to is the bone, and to sit there for decades, 10 years, 20 years, 30 years, totally invisible to any methods we have of scanning. I mean, you can go and take out pieces of the bone, but you're not going to do that when someone doesn't have a disease that's obvious. So you go into total remission for 20 years. 20 years later, you come back into the clinic and are diagnosed with metastatic bone cancer from the original cells that were deposited there in your breast cancer 20 years earlier. And the question is, what are those dormant cells? What's going on? So people study this just like they study persisters. And it's sort of similar from my perspective. So here is my table of similarities and differences. There's stresses. In the drug resistance, you have stress because you're interfering with some oncogenetic pathway like the EGFR system. In dormancy, you have stress because you've landed in a foreign environment. Some cells persist, some cells go dormant, most of them do not. There's all sorts of ways in which those cells are similar. And the one thing which I think is very different, there's also metabolic correlates of dormancy. The thing that's very different and the thing which we're trying to understand is persisters can seem like they can transition back to cycling with no extra perturbation. It seems like a natural thing that they're able to do via their own dynamics, whatever that is in detail. Dormant cells awaken, that's why you get metastatic bone cancer, but it seems like there's always some external trigger. It seems like they cannot just by a noise or fluctuations just awaken. They actually need to have some signal that awakens them. And people have been studying, again in some most recent Nature paper, how various, for example, viral infections creating inflammation can awaken dormant cells. So it seems to be that there's a very big difference in terms of, one is sort of almost an inevitable consequence of the cell trying to adapt, and as long as they can last long enough, they get there. In the other case, it really requires an external trigger to do, at least so. But why? Who knows? And finally, just to close the loop on everything, this appeared very recently as a bioRxiv posting. This is a very well-known immunotherapy lab. And their immunotherapy lab, this is the first time that I know of that people have identified the phenomena of persisters for immunotherapy. And so persisters is where some type of phenotypic change has somehow not prevented the drug from acting on you. It still acts on you, but somehow you go into some state which has no cycling, has sort of a quiescent metabolism, and nonetheless you survive, you don't die. And these people identified that this can happen in immunotherapy, and this is their very interesting experiment. They did melanoma cell lines, 3 different types. These cell lines are known to be targets of various T cells, so you can add in a culture system, add adaptive immune cells that specifically target those melanoma cells. And in terms of these 3 things, and they looked for survival, based on that system, how many cells survive the attack by the T cells that they know can recognize those as being foreign objects. So they remove that part of the problem. They take the T cells that they know can attack these. And the answer is that in their 3 things, 2 of them had signs that the attack happened and they just avoided dying. And one of the ways they avoided dying is they downregulated the apoptotic pathway in this system. And the other one had the more traditional way in which you can immune immunotherapy. The T cell was detecting this particular antigen, and the cells just mutated away that antigen. That's the traditional way in which you would get rid of immune attack, is you would drop the antigen, and then, okay, do you need that antigen? Is a really an important part of your growth cycle. That's the challenge. But here they showed that there were at least ways in which you can get around it that were more analogous to persisters rather than to pure resistance. And again, some of the features they talk about for these again follow some of the similar rules. Okay, So we're done. So cancer cell plasticity is a critical component of many aspects of tumor progression. I've tried to give you some perspective on that from the point of view of the drug resistance. We're studying a particular model, and we can explain qualitatively or semi-quantitatively various aspects of this. There's a ton of more work that we have to do, and this was originally work I started with my longtime collaborator, Dave Kessler, and now is being continued by a student, Jitae Park, who has the advantage of having a strong pharmacology background, so he can actually read papers on things like metabolism and epigenetics and understand them. And we're trying to sort of finish up our preliminary calculations that I showed you some of in today's talk. And thank you very much for your attention.